A GMO is a genetically modified organism. Modification occurs in a laboratory where a scientist inserts DNA from one organism to another. The purpose of creating GMOs is to strengthen the host organism with traits that will aid survival.
My name is Nidhi Rawat and I am an assistant professor of plant pathology at the University of Maryland. When we are trying to make a GMO, our first step is to identify a gene that interests us, that has the properties of getting transferred and still expressing the good trait. So the first step in creating a GMO is to find a gene that is useful for us. It can be a disease resistance gene, it can be a herbicide resistance gene. For example, in soybean we have GMOs that are resistant to herbicides, so we can get rid of the herbs in the field or weeds in the field by spraying them with a particular chemical but not killing the plants themselves. Our next step after we identify the gene is to transfer it. One of the ways is using a bacterium that's called Agrobacterium tumefaciens. So we introduce the gene into the body of the genome of the bacterium, and then we infect the plants which we want to create GMO of with that engineered Agrobacterium tumefaciens, which then transfers the desired gene into the plant and hopefully expresses itself.
Using a gene gun, the DNA or transgenes from desired plants are shot like bullets into the callus cells of the host plant. These DNA strands are also known as gold particles because the injection shoots small metal particles coated in DNA. This process is also known as the biolistic particle delivery system. This method can also be used to change any other part of the plant's cell structure, including the nucleus and the organelles.
The next step is to test if it is working, if it's expressing itself there, and if it is actually present through the generations. So this step is done in multiple generations, but the first step is to see using PCRs or biochemical assays to make sure that the gene is in the genome now. After the gene is integrated into the genome and we check that it's there, the next step is to use it actually for the public consumption. But before it comes to the market, it has to be tested for its non-interference with any other system of the ecosystem.
To learn more, visit the National Center for Biotechnology information at nih.gov.
GMO, now you know.